Hi, my name is Valerie Gouon-Evans. I'm from Boston University, and I would like to thank the organizer for giving me the opportunity to share with you my lab's work on VEGFA promoting biliary epithelial cell to hepatocyte conversion to treat acute and chronic liver disease. So 6 to 8 million patients suffer from a chronic liver disease in the USA. So based on the underlying cause of the disease, up to 46% of the American population is diagnosed with non-alcoholic fatty liver disease, or MASLD, that can then progress to liver fibrosis and liver cirrhosis and eventually cancer. So the current treatment focuses on eliminating the underlying liver insults, and liver transplantation remains the only treatment for end-stage liver disease. Therefore, there is an urgent need for alternative treatment to prevent progression and reverse the liver disease. Acetaminophen overdose is a leading cause of liver failure in the US. It is what the human liver histology looks like after poisoning. It's covered with necrotic area, shown here in pink color. And acetaminophen overdose accounts for about 50,000 emergency room visits per year. And importantly, 30% of liver transplants in the US. So the only treatment is N-acetylcysteine, and it is efficient when administered the first 10 hours after poisoning. So because most of the patients present to the hospital late, liver transplantation becomes the last solution, which is challenged by the lack of donor organs. Therefore, again, there is an urgent need for discovering treatment with a wider therapeutic window to prevent liver failure and minimize liver transplantation. So to address this clinical gap, my lab develops strategies to harness liver regeneration by promoting the naturally occurring repair mechanisms to generate healthy hepatocytes, the most functional cells of the liver. So the liver has a fascinating ability to regenerate this hepatocyte cell mass. But the first and most efficient mechanism is simply via proliferation of mature hepatocytes, so hepatocyte-driven liver repair. However, in case of severe acute or chronic injury, hepatocyte proliferation is compromised and they become senescent. In this case, the cholangiocyte-driven liver repair mechanism can take over. In this scenario, some cholangiocytes are believed to be a source of liver progenitor cells, and as such, they proliferate and differentiate into hepatocyte. This self-conversion have been first described in rat in 1989 by Sorgensen's group, then in human by the observation of transitioning hepatocyte co-expressing cholangiocyte marker and budding from string of cholangiocyte in ductal reaction of advanced liver disease. And then the self-conversion was formally demonstrated in cholangiocyte lineage tracing animal models such as zebrafish, where hepatocytes were severely ablated or chronically depleted. And finally, in mouse model, the conversion was shown significant when hepatocyte proliferation was experimentally compromised by, for example, overexpression of P21, mimicking a hallmark of the chronic liver disease, or by depletion of beta-catenin in hepatocyte, or by inducing long-term chronic liver disease. For this repair mechanism to become a potential therapy, it needs to be harnessed. So today I will tell you the project from Fatima Rizvi, a postdoc in my lab, who demonstrated that VEGFA harnesses this cell conversion by 5-fold and reverse some feature of the chronic liver disease. So to deliver VEGFA to the liver, we decided to use nucleoside-modified mRNA complex into lipid nanoparticle. So nucleoside-modified mRNA are in vitro transcribed messenger RNA, and they harbor modification pioneered by Nobel laureates Katalin Karikó and Drew Weissman. And this modification prevent immunogenicity and increase mRNA stability and translation into protein. And they have many advantages. They are clinically safe as they do not integrate into the genome. And when mRNA are complex with lipid nanoparticles, they specifically target the liver. And they allow control expression of protein with time, a week at most, which is a key advantage when we induce mitogen expression. So all mRNA LNP used in our studies were provided by Drew Weissman and Norbert Pardy in collaboration with Ying Tam from Acuitas. So first we injected in mice luciferase mRNA LNP to confirm the liver specificity of mRNA expression. And that expression lasted about 3 days. So to identify the cells that are transfected in the liver, we injected EGFP mRNA LNP. And we found that virtually all hepatocytes, the most functional cells in the liver, were transfected, as well as 70% of endothelial cells and 40% of the Kupffer cells, the macrophage of the liver, while the cholangiocytes were not transfected at all. So what was the rationale for using VEGFA to promote cholangiocyte to hepatocyte conversion? So VEGFA is the ligand for the receptor VEGF receptor 2, or also called KDR. And we previously showed that KDR is expressed on hepatic progenitor cells using human and macaque pluripotent stem cell hepatic differentiation culture. Importantly, in this culture, inhibition of KDR decreases the efficiency of this progenitor to differentiate into hepatic cells. This suggested that activation of the KDR, the receptor, promotes hepatic differentiation. And we also identified KDR expressing progenitor in the developing mouse liver and show that they could give rise to hepatocyte in adult mice. So this finding led us to speculate that the regenerating liver uses the same cues as the developing liver. And Fatima specifically asked, is VEGF receptor 2 expressed on cholangiocytes, which are a source of progenitor cells following liver injury? And then can we use then VEGFA to activate cholangiocyte to differentiate into hepatocyte and rescue liver function? So in the liver, KDR is expressed on sinusoidal endothelial cells. And those are seen here in green and here in red. However, KDR is not expressed on cholangiocyte in homeostasis condition when the liver is not injured. But in contrast, in response to both chronic and acute liver injury, we observe a subset of cholangiocytes that express KDR. And they are indicated here with the arrowheads in this both condition. So to test her hypothesis, Fatima used both acute injury, which is an overdose of acetaminophen, and the chronic CDE diet liver injury, which is the choline-deficient ethionine supplemented diet. And mice were injected with AAV P21 to overexpress P21 in hepatocyte and thus mimic hepatocyte senescence, which is observed in virtually all human liver diseases. So VEGFA was delivered to the liver with mRNA LNP. And as you can see, VEGFA serum levels are very high by the first day after injection of mRNA LNP. And then they drop very drastically the following day. So she lineage traced the fate of cholangiocyte using inducible Cree tomato reporter mice. In this model, upon tamoxifen injection, cholangiocyte become tomato positive, red, as well as the hepatocytes that are generated from them. So in the following slide, we will look at tomato-positive hepatocyte, meaning that they would come from cholangiocyte. So this is the result of the acute injury. Here, mice were administered acetaminophen, followed by 2 injections of VEGFA, mRNA, LNP. In absence of VEGFA, rare tomato-positive hepatocytes were visible. Here you can see one. However, VEGFA drastically and significantly induced differentiation of cholangiocyte into hepatocyte. And that was quantified, and we found that it was about 5-fold compared to control injected mice. So you can see, and the identity of this hepatocyte that we can see here, large cluster. The identity of this hepatocyte was confirmed with expression of the transcription factor HNF4 alpha. And this cell conversion was very significant and occurred in all liver lobes in each mouse as quantified here, with a 5-fold increase in the presence of VEGFA. So importantly, in the chronic liver disease now, so the CDE diet injury model, VEGFA not only promotes cholangiocyte to hepatocyte conversion about, again, 5-fold. As you can see here, this large cluster of hepatocyte in presence of VEGFA, while this cluster were very small actually in control treated mice. But not only we see this self-conversion, but also what we found is that VEGFA completely reverted steatosis, which is seen in control in green, and also reverted fibrosis, which is seen in blue in control, and that was very significant. So to investigate the mechanism of action of exogenous VEGFA delivered with LNP, Fatima used the cholangiocyte brain bow reporter lineage tracing, allowing us to track single clones of GFP-positive cholangiocyte. That was examined in the chronic injury, the CDE diet, 5 days after a single injection of VEGFA or control mRNA LNP. So we found that VEGFA drastically induced emergence of GFP-positive clusters that lost the cholangiocyte marker EpCAM, indicated in pink here. And those cells are indicated with the arrow. And this supports the role of VEGFA in dedifferentiating cholangiocyte, and that was very significant. As expected, we found that some of the cells, the GFP-positive cluster, also express KDR, as you can see here with this arrow. And virtually all cells within the GFP positive cluster are proliferative, as seen here with EDU co-staining with a GFP. So EDU in pink and GFP in green. So supporting further the role of VEGFA in promoting dedifferentiation of cholangiocytes that are highly proliferative. So for potential clinical translation of VEGFA mRNA LNP, we looked for KDR expression in cholangiocyte in advanced human cirrhotic specimen, and that was done in collaboration with Alex Soto Gutierrez from Pittsburgh University, who provided us with the specimens. So to our surprise, we identified at the margin of the regenerative hepatocyte nodule. So on that side you have the hepatocyte nodule, and on this side there is a fibrotic septa where cholangiocyte proliferate, and it's what we call the ductal reaction. So in this ductal reaction area, we could find some cholangiocytes that co-express KDR. So for example, here if we look at the frame 1, we see a lot of KRT7 cholangiocytes that are also KDR positive. We also found some intermediate hepatocyte expressing KDR that are budding from this KDR-positive cholangiocyte, and even some large cluster of hepatocytes that express KDR, just at the merging of the regenerative nodule. So we further validated KDR expression at the transcript and also the protein level in purified hepatocytes from 9 human advanced cirrhotic specimens compared to 3 normal specimens. And this was done again in collaboration with Alex Soto-Gutierrez, who provided us with all human specimens and purified hepatocyte. So KDR transcript as well as protein levels were upregulated in purified diseased hepatocyte compared to normal hepatocyte, seen here for the Western blot or here for the RT-qPCR. So this data suggested that VEGFA mRNA LNP may serve as a therapy to potentialize cholangiocyte-driven regeneration in advanced human liver disease. So in conclusion, based on the literature and our work using various animal models and human specimens, there are clear evidence that cholangiocyte to hepatocyte conversion occurs when hepatocyte proliferation is compromised, which is not surprising as hepatocyte and cholangiocyte come from a common progenitor, the hepatoblast, that develops in the fetal liver. And not only they can self-renew hepatocyte and cholangiocyte, but they can also replace each other when needed during injury. And there are some good evidence also that the cholangiocyte to hepatocyte conversion is mediated through a liver progenitor cell that is highly proliferative. And that was demonstrated in zebrafish and in mice and in our studies that I just showed you. So today I showed you that VEGFA delivered with mRNA LNP can promote this conversion fivefold to potentially consider this mechanism as a therapeutic means to alleviate the liver disease. So I would like to acknowledge my whole lab, who established as a team the mRNA LNP platform to promote liver repair, and especially I would like to acknowledge Fatima Rizvi, who did the work I presented today. I would like to thank our collaborators, Drew Weissman, Norbert Pardy, and Yin Tam, who provided us with all mRNA LNP used in our studies, and as well as Donggun Chin and Alex Soto-Gutierrez, who are our collaborators on this project. And here our funding source. So thank you so much.